in terms of weight training specifically, could you talk to us a little bit about some of the biggest genetic factors that we see that will affect somebody's ability to grow muscle?
Yeah, so one of my lab's interests, and this is what Cody Hahn was doing when he was a PhD student here, was trying to determine molecular factors, if you will, that differentiate high anabolic responders versus individuals that really don't grow muscle in response to weeks or months of resistance training. And I have to give credit to Dr. Marcus Baumann, who's actually at UAB, so he's in Birmingham down the road. He was really the first researcher that started addressing this question. So a lot of our studies were really inspired by his original studies in the mid-2000s. But the idea is that we take a group of people, and we've done 2 of these studies now, so we've taken 2 groups of people, and we train them for 6 to 12 weeks in the weight room. Everybody gets the same relative load according to their strength. And we do pre and post intervention assessments, and we look at changes in things like muscle thickness using an ultrasound. Now, some people, they'll see this robust increase in muscle thickness, or, we also take biopsies out of the leg, and they'll show this increase in muscle cell size. So we call those the high responders. What's fascinating to us is that some people show no meaningful response, right? So those are your low responders. From there, since we have the tissue, what we try to do is we try to look at biomarkers that exist at baseline that could be explaining, right, why one group responded whereas the other didn't. And then we also look at the pre to post change in those biomarkers in muscle. And so we haven't done formal genetics testing. Like, you can probably go onto PubMed and look up genome-wide association studies for any particular trait like BMI. That's where the FTO SNP, the obesity risk allele, pops up if you look at BMI and genome-wide association studies. But the idea there is that researchers will get a lot of people, a lot more than just 100 people. They'll get thousands of people, and they will test all of these polymorphisms from a blood sample or a saliva sample from buccal cells, and they'll say, these genetic variants are linked to the phenotype. So we haven't done that, but what we have done is we've looked in muscle, and we found a couple of biomarkers which may be suggestive of why some people respond to training versus why others don't. And the main marker in muscle seems to be ribosome content. Mm-hmm. So for listeners that aren't aware of the physiology there, ribosomes are the macromolecules which facilitate muscle protein synthesis. So to us, that kind of makes sense. If you see an increase in what's called ribosome biogenesis, or the creation of new ribosomes with resistance training, then according to our data, as well as others like Marcus Baumann, you probably will be a high responder. Whereas if you don't see an increase with training, then you may not be a high responder.
Yeah, it's interesting because, anybody who's taking like cell biology, and, we all have that like generic idea of the cell. And it was never really taught that there would be variations in the number of organelles that are in there. I mean, it makes sense, like there's variations in pretty much everything. Sure. But to think that one factor can have a significant impact is pretty interesting. Yeah. I'm curious, what is the longest study you've seen where somebody was not responding at all? Because a lot of these are shorter term.
Yeah, they are. Yeah, so that's a good point, Dave. I think Marcus's studies, his were 4 months in duration.
Okay, that's decent.
Yeah, that's pretty decent. Our 2 studies, so Cody's study was 6 weeks. Now, keep in mind, he recruited previously resistance-trained individuals for his study. And then our other study was done by Brooks Mobley, who's now at Kentucky, but we did a 12-week training study there. So that's a fair point. Cody's study in particular, 6 weeks, one can make the argument that, well, perhaps the low responders may have started responding after that time point.
Right.
We think 3 to 4 months is a good amount of
Yeah.
time. And I tell people, look, because we get a little bit of pushback on this, but like everything else, I mean, it's a bell-shaped curve in terms of the respondents, right? You're going to have people that are there towards the far right, which are your extreme responders. You're going to have a lot of people in the middle, which are going to be kind of your mediocre responders. And then there are always going to be people that are there towards the left end of the curve, which are your low responders. I mean, that's pretty much how everything works. So
Right.
to us, it's not shocking that we see that. We're just trying to understand the molecular reasoning behind that, right?
Sure. Have you looked into anything with the androgen receptor density?
Yeah. So I was getting back to, other genetic components which may delineate responders versus non-responders. So besides Marcus's data and our data, Stu Phillips has done a lot in this area. Rob Morton's his PhD student, and he recently put out a paper saying that if you have a high level of androgen receptor in the muscle, you will be a high responder. And I believe that study was in previously trained males as well. So it was like Cody's study. So we actually tried to replicate that assay. We did Western blotting, and we validated our antibody through the appropriate means, for lack of a better term. We didn't see anything similar to what Rob and Stu reported. That's not to say that they're wrong. It's just, at least in our cohort, the androgen receptor doesn't play a role. And that is for both studies, right?
Wow.
So the relative amount of androgen receptor, whether you're untrained or trained, doesn't drive the anabolic response, according to our data. Rob's paper seems to suggest that it does. So, like all science, right, you're going to have divergent findings. I think it's something that definitely could be contributing. We just need to try to figure out, if it's a thing or not with more research.
Yeah, that's interesting. I had thought most of the studies on it had shown that androgen receptor density was a pretty important factor. I didn't actually realize that these two recent ones show that it was not a factor.
Yeah, so the only Stu has, I think, 2 papers now one was showing that the change in androgen receptor positively associated with the change in muscle hypertrophy. And that was by Cam Mitchell, I think, and +1. And then Rob's most recent data, they actually compiled a bunch of participants from multiple studies, and they showed that both at the pre-intervention time point as well as the post-intervention time point, that the high responders had more muscle androgen receptor.
Right.
Our data doesn't say anything to that tune, right? So,
Yeah.
in study number 1, which is 12 weeks untrained people, in both the high and the low responders, we saw a decrease in the androgen receptor protein with training. Okay. And we said, well, to us, this kind of makes sense, because if resistance training is operating in part through testosterone signaling in the muscle to facilitate adaptation, then there could be this compensatory downregulation in the androgen receptor, right? You increase the signaling, you're going to potentially downregulate the receptor.
Right. Interesting.
In study number 2, which is Cody's study, we didn't see any change. And so then from there, we bought a positive control, which was a cancer cell lysate. It was a prostate cancer cell lysate. We did a Western blot on that to make sure that our antibody was valid, right? Because that's critically important with Western blotting. And it indeed was valid. So we also supplied that with Cody's data saying, look, we understand that some people are saying this, that androgen receptor protein levels are important. We're not seeing it. Now, that's not to say that signaling, there's a difference between looking at the protein content of a receptor in muscle versus looking or interrogating how that protein functionally operates, right? So I think to test that question, you would have to design a study that is long-term to say these are responders, these are non-responders. You get like a pre and a post-intervention biopsy. But then there at the very beginning, you perhaps do a bout of training and you get a kind of a series of biopsies following that first bout of training, because from there, you can look at the androgen receptor in terms of where it's localized, right? Does it move to the nucleus to enhance its effects on the genome, right? Because that's how androgen signaling operates. It binds as a transcription factor to turn on genes related to cell growth. So I think you need to do that to really start parsing out the importance of the androgen receptor in terms of the hypertrophic response.